Hi all, we have learned how urine is formed in kidneys. So this urine formed in the kidneys will be collected in the urinary bladder and get stored there. Initially there will not be much rise in the intravesical pressure in the bladder, but as the urine gets accumulated, this pressure will increase, and when it reaches a suitable level, this urine will be emptied out. So, the process by which urine is voided from the urinary bladder is called micturition. And this micturition is a reflex process, but it can be voluntarily controlled to an extent by the adults and grown-up children. In babies, this control on urination starts to develop at the age of 2 years and gets completed by 3 years. So let's see how this micturition happens. Urine is formed continuously by the nephrons and it flows down into the bladder drop by drop through the ureters due to gravity supported by peristaltic movements. When urine is collected in the renal pelvis, the contraction sets up there and it is transmitted through the rest of the ureter as peristaltic waves. So these peristaltic waves originate by the pacemaker tissue at the renal calyx at a rate of about 1 to 5 per minute, and it travels at a velocity of about 3 centimeters per second. It reaches up to the trigone of urinary bladder. This peristaltic wave pushes the urine into the bladder. And at the bladder, there is a valvular arrangement at the point of entry of ureter into the bladder. And also this ureter turns horizontally before entering the bladder. So this valvular arrangement at the end of ureter and the position of ureter prevent the backflow of urine from bladder into the ureter when the bladder contracts. The functional aspects of urinary bladder. Urinary bladder is a triangular, hollow, sac-like structure present in the lower abdomen. It is a distensible organ made up of the smooth muscle, detrusor muscle. The term detrusor implies that pushes down. So the contraction of detrusor helps in the emptying of bladder. When we look into the nerve innervation, we can see that the bladder is innervated by sympathetic and parasympathetic fibers. So first the sympathetic nerve supply. The preganglionic fibers of the sympathetic nerve arise from the first and second lumbar segments of the spinal cord, that is L1 and L2 of the spinal cord, and they leave the spinal cord, passes through the lateral sympathetic ganglia and terminates in the hypogastric ganglion. There you can see it in the picture. So after leaving the spinal cord, the nerves that arise from the first and second lumbar segments, those nerve fibers will terminate in the hypogastric ganglion. And from the hypogastric ganglion, the postganglionic nerve fibers arise and form the hypogastric nerve. This hypogastric nerve supplies the detrusor muscle of the bladder and the internal sphincter. The function of this hypogastric nerve is that it causes relaxation of detrusor muscle and constriction of internal sphincter. So what happens when the detrusor of the bladder is relaxed and the internal sphincter is constricted? It helps in the filling up of bladder. Okay, so this sympathetic nerve, that is the hypogastric nerve, is called as the nerve of filling. Now, the parasympathetic nerve supply. The preganglionic fibers of the parasympathetic fibers arise from the second, third, and fourth sacral segments of the spinal cord. So the S2, S3, and S4 segments of the spinal cord, they form the parasympathetic preganglionic fibers, and these fibers form the pelvic nerve, and this pelvic nerve fibers pass through the hypogastric ganglion and synapse with the postganglionic neurons, which are situated nearer the detrusor and the internal sphincter. The function of this parasympathetic fiber is that, parasympathetic nerve is that It causes contraction of detrusor and relaxation of internal sphincter. That is the function opposite to that of hypogastric nerve. Hypogastric nerve helped in the relaxation of detrusor and contraction of internal sphincter. Here, the pelvic nerve helps in the contraction of detrusor muscle and relaxation of internal sphincter. So what happens here when there is parasympathetic stimulation? There will be contraction of detrusor muscle and relaxation of internal sphincter, which means there will be emptying of urinary bladder. Hence, this pelvic nerve is called as the nerve of emptying or nerve of micturition. And this pelvic nerve is also having a sensory part. There are stretch receptors in the walls of urinary bladder and urethra. So when the bladder and urethra are filled with urine, these stretch receptors will sense it, and the impulses from these receptors will be carried to the central nervous system via the pelvic nerve. Okay, so that is the sensory part of pelvic nerve. Then comes the somatic nerve supply. Here the external urethral sphincter is innervated by the somatic nerve called the pudendal nerve. And this pudendal nerve arises from the second, third and fourth sacral segments of the spinal cord, that is S2, S3 and S4. And the function of these somatic fibers is that it maintains the tonic contraction of the skeletal muscles in the external sphincter. That is, it keeps the external urethral sphincter always constricted. Okay, so with the help of this pudendal nerve, the external urethral sphincter always remain constricted. And during micturition, what happens is that this pudendal nerve will be inhibited. So what happens? The pudendal nerve helps or keeps the external urethral sphincter in a constricted state, and when this nerve is inhibited, what happens? There will be relaxation of the external sphincter leading to voiding of urine. So the pudendal nerve is responsible for the voluntary control of micturition. Okay. Usually the external sphincter remains constricted because of the help of this pudendal nerve, but during micturition this pudendal nerve will be inhibited so that the external sphincter will be relaxed and helps in voiding of urine. So this pudendal nerve is called as or is responsible for the voluntary control of micturition. We have seen that the urinary tract is having two urethral sphincters, the internal urethral sphincter and the external urethral sphincter. The internal urethral sphincter is located at the neck of the bladder, that is in between the neck of the bladder and the upper end of the urethra, and is made up of smooth muscle fibers. It's innervated by both sympathetic and parasympathetic nerves, that is hypogastric and pelvic nerves, and hence it is under autonomic control. And the external urethral sphincter, it is located in the urogenital diaphragm, that is around the urethra in its proximal part, and is formed of circular skeletal muscle fibers. And this external sphincter is innervated by somatic nerve, that is the pudendal nerve, and therefore it is under voluntary control. There are two main functions for the urinary bladder. First one is the storage of urine up to a critical volume. And second is when this critical volume is reached, the bladder will empty the urine into the urethra. So these are the two functions served by the bladder. And when the urine is filled in the bladder, that is when the bladder volume increases up to 150 ml, we will have that feeling of bladder filling. And when this volume reaches about 150 to 250 ml, we will have the sensation to pass urine. And when this volume reaches about 400 ml, this sensation to pass urine becomes uncomfortable. And when this volume reaches about 700 ml, it becomes very painful, leading to the loss of control. Micturition is the process of passing urine or voiding urine. It is a reflex phenomenon. The centers are mainly located in the spinal cord but under the influence of higher brain centers. When urine is collected in the bladder, the pressure inside the bladder increases. The pressure inside the bladder is called intravesical pressure. Initially, when there is accumulation of a smaller quantity of urine, there will be a rise in the intravesical pressure and it remains unchanged until the volume of urine accumulated reaches a particular level. That is, there will be an initial increase in the intravesical pressure when there is a slight accumulation of urine, and this intravesical pressure remains the same until a particular quantity of urine is collected in the bladder. And this is because of the property of the smooth muscle in the bladder wall called plasticity. And plasticity of a muscle is the ability of that muscle to alter its structural and functional properties according to the environmental conditions. So, initially, when the urine collected in the bladder, say it's about 50 ml, there will be an initial rise in the intravesical pressure, and that pressure remains almost the same until the bladder gets filled with urine. So, when the bladder gets filled with urine, there will be a sharp rise in the pressure that initiates the micturition reflex. So, when the bladder gets filled, the stretch receptors in the bladder wall are stimulated and sensory impulses are generated that are carried to the central nervous system via the afferent nerve fibers of the pelvic nerve. Okay, the sensory part of the pelvic nerve that we have seen earlier. Now, the micturition reflex. We have seen that the urge to pass urine is initiated when the bladder gets filled and is sensed by the stretch receptors in the bladder wall. So, when the stretch receptors are stimulated, the afferent impulses are generated and they are carried by the afferent or sensory nerve fibers of the pelvic nerve, and these impulses reach the sacral segments of the spinal cord. There, in the spinal cord, motor or efferent impulses are generated and these impulses are carried by the motor fibers of the parasympathetic pelvic nerve to the bladder and the internal sphincter. We know that the motor fibers of the pelvic nerve cause contraction of the detrusor muscle and relaxation of the internal sphincter. So this causes flow of urine from the bladder into the urethra, and once the urine enters the urethra, the stretch receptors in the urethra are stimulated and send afferent impulses to the spinal cord via the pelvic nerve. So now the motor impulses are generated in the spinal cord that causes inhibition of pudendal nerve. We know that pudendal nerve keeps the external urethral sphincter constricted. And when the motor impulses cause the inhibition of pudendal nerve, it results in the relaxation of external sphincter that finally results in the voiding of urine. Okay, so this is what is happening in the micturition reflex. When the stretch receptors of the urethra are stimulated, the afferent impulses will be carried by the sensory part of the pelvic nerve. See, here pelvic nerve is having both sensory and motor part. The sensory part of the parasympathetic pelvic nerve carries impulses from the stretch receptors to the central nervous system, and the motor part carries the impulses from the central nervous system or from the spinal cord to the bladder and the sphincter, internal sphincter. So it causes the contraction of the detrusor and relaxation of the internal sphincter, resulting in the entry of urine into the urethra. and the stress receptors there will be stimulated. See, here when the urine enters the urethra, we can voluntarily control the micturition by the contraction of the perineal muscles and the external sphincter. Okay, by the contraction of the perineal muscles and the external sphincter, we can voluntarily control the micturition. If otherwise, that is, we are unwilling to control, what happens? There will be inhibition of the pituitary nerve, resulting in the relaxation of external sphincter, and finally, micturition happens. Okay. One important thing to be noted is that once a micturition reflex begins, it is self-regenerative. That is, the initial contraction of the bladder further activates the sensory receptors to cause still more increase in the sensory impulses from the bladder and urethra, and these sensory impulses in turn cause further increase in the reflex contraction of bladder. So this cycle continues repeatedly until the force of contraction becomes maximum and the urine is voided out completely. Another thing is that this micturition is a parasympathetic activity. The sympathetic nerve fibers are not having a role in micturition. Only the parasympathetic nerve fibers are playing a role in micturition. And we can see that during micturition, the flow of urine is facilitated by the increase in abdominal pressure. See, the contraction of abdominal muscles and descent of diaphragm increases abdominal pressure. And this increase in abdominal pressure facilitates the urine flow during micturition. Not only increase in abdominal pressure, but relaxation of the pelvic, perineal and anal muscles also facilitate the flow of urine during micturition. Then we have already said that the contraction of the external sphincter and the perineal muscles can voluntarily control micturition. That is, when the urine enters the urethra from the bladder, we can voluntarily control this micturition by contracting the external sphincter and the perineal muscles. So, another thing is that the cortical activities and learning to contract the external sphincter can influence our voluntary control in micturition. That is, the brain cortex is having a voluntary inhibitory control on micturition. That is, it is having a inhibitory effect on micturition which can be voluntarily controlled. And in children below 3 years of age, this cortical activity is not so developed that they often pass urine without their knowledge. So that is happening in children under 3 years of age. So these cortical activities and learning to contract the external sphincter can influence our voluntary control on micturition. Okay. Role of higher centers in micturition. We have seen that the micturition reflex is having its integration centers mostly in the spinal cord, but is also under the influence of higher brain centers. So the higher centers which control micturition are of two types: facilitatory centers and inhibitory centers. Facilitatory centers are the centers that facilitate micturition via the spinal centers. Pons and posterior hypothalamus are having facilitatory area. Inhibitory centers are those that inhibit the micturition by suppressing spinal micturition centers. Midbrain is having an inhibitory area. Likewise, cortex is having a voluntary inhibitory control on micturition that we have said already. So the voluntary control in children starts to develop at about 2 years and completes by 3 years. Thank you.